Welcome to this tutorial on using a microscope and a camera attachment. These are pieces of equipment that you're going to need to be able to use in order to be successful in the stomatal density lab, which is a two-part lab. We have a set experiment that we're going to be looking at slides, and then you're going to design your own independent research questions. So pay attention. The basic idea of the lab is we're going to get leaf material from plants growing in different environmental conditions. We're going to make some impressions of the undersurface of those leaves where the stomata density is highest. And then we're going to use microscopes to actually look at the stomates themselves. And here we are with a red maple in the field, making impressions. And each one of these little openings here is a stomate. Stomates are used for gas exchange, and there's a lab video that will help you explore those ideas. What I want to do with you here is really just talk to you about using a microscope. So here's a diagram that shows you the parts of a microscope, and here following is a YouTube video that I put together to explain using a microscope.
Okay, hello, and welcome to Ecology Lab, where we're going to think a little bit about microscopes. Microscopes are a great way of seeing things close up, imagining life in ways that we can't imagine with our naked eye. You've all used microscopes before, and making sure you remember how to use a microscope is going to be key to being successful in this stomatal lab, which is going to be a 2- or 3-part lab for you. So we're just going to go over microscopes. So you know that a microscope has 2 eyepieces that you look through. These are binocular microscopes. These are called oculars. They have their own level of magnification, and they are attached to the headpiece, which then attaches to the objective lenses. These objective lenses rotate. They have different levels of magnification. So the smallest level of magnification is 4x. So that's enlarging it 4 times its natural size. The ocular pieces are each 10x. So you multiply the 2, that's 40 times larger than you could see with your naked eye. If you rotate it to 10x, that's 10. Increase of magnification by 10 and 10 here. So that's 100 times more powerful. And the last one that we will use for our lab is the yellow one, which is 40x. And so 40 and 10 is 400 times. And that's the magnification in the end that you're going to take your data from, okay? Whenever you start looking at a slide, you will look at a slide starting with the 4x objective lens. So let's say that you've actually made a slide. So here's a slide that I've made. You're going to put it into the stage, and I'm going to bring this a little bit closer so you can see the stage. You want to have it placed within the stage on both sides so that when you move the stage arm back and forth, it will move the slide, and that allows you to look at different places within the slide itself. Okay, we're back again. So if you were going to look at this, you would want to turn the light, the microscope on, and there's a little on switch that's down there at the base of the microscope. And there's a couple other things I want to show you. Underneath the microscope here, there is a condenser which changes the color of the light that gets through. And there's a diaphragm that changes the intensity of the light that gets through. So we're going to actually come up and look at that. Just look at how that gets brighter, and that would probably burn your eyeballs out, so be careful with that. I often work on it very low intensity. And the last thing you should recognize is there actually is a way of making the light itself brighter as well as the diaphragm, the amount of light that's allowed to come through. Okay, so those are the primary pieces of the microscope that you need, that you'll be working with. The 2 ocular pieces you can move further apart or close together. That'll help you with the distance between your eyes. And to focus, there's this big knob, that's gross focus, and there's a little knob, which is fine-tune focus. So when you start to work with a slide, you're going to work with it at 4x. Remember, 4 times 10 is 40 times magnification. You get it in focus at 4x, and make sure it's nice and clear for your eyes using the gross focus. And then you would switch over to 10x and work your way up. Now, if you've been paying attention, one of the things that you've noticed is there were actually 4 objective lenses. We do not work with the last one. The last one is actually 100x. So 100 times 10 is 1,000. So that would give you 1,000 times of magnification. To use this last objective lens, you actually have to put a drop of oil on your slide. We are not going to do that for this lab. Okay, so just to review, you've got 2 ocular pieces. They're binocular. You adjust them for your eye width. You start out at 4x. You use your gross focus to bring it into focus. You can fine tune with a little knob. And you rotate up through your objectives. To take the slide back off, you put the lenses, so no one lens is down. You open up the stage clip and you remove the slide. And that, my friends, is how to use a microscope. If you have questions, we can go over them in class.
Okay, so once you have got your slide and you've worked with your microscope, you're going to recognize it's going to take some patience. Getting something into focus, gross focus, fine-tune focus, and working with that diaphragm to allow light in and out. But ultimately, you're going to want to get some up-close pictures of stomata. So here is the stomate at 400 power from a mother-in-law tongue. This is taken during the day, and you can see that the guard sails are nice and tightly closed. And here's a different species of plant, some kind of stem cactus, and this is at nighttime, and you can see those guard cells are opened up, and that plant is actually going through exchange of gases. So you're going to actually need to get your stomata on the undersurface of your leaves at 400x in clear enough magnification, and you're going to want to take a picture of one stomate because you're going to take a measurement of the width of that stomate. So we will watch a short YouTube video on how to use the camera.
Hello again. So this short video is a tutorial on how to use the camera in association with the microscope so that you can collect images of your stomata. All right, so I'm going to rotate this a little bit so we can see that computer. You remember how to use a microscope. So I actually have a prepared slide. This is going to be one of the slides that's going to be in your pack that you're going to be working with. And I'm going to try and get images of these stomata, and I actually want to try and measure these stomata. So we're going to do that using a camera. So there is a camera app here, and what you need to do is you need to carefully remove one of the ocular pieces. You need to place this ocular in the cushioned the cushion that's provided by your microscope. They're very expensive. We don't want to mess that up. You then take the cap off of the camera adapter and you put the camera adapter in that ocular piece. You can then see that there is an attachment from that to the computer. You are going to first need to download the software for the Amscope onto your laptop, and that will be available for you to do in lab. You then just plug this into that eyepiece, that camera, and it goes into a USB port on your computer. We have the lovely little ringy dingy. And now we're going to rotate this around and I'm going to show you the camera. Okay, so this is what it's going to look like, and you're going to have Microscope, camera list, and we're going to go ahead and click on MD500, and that's this camera. And it's going to show me, eventually, an image of what is under the scope. And this is only a fraction of what you're going to see. But you can see that there are some images, and I'm going to actually bring those into focus. And what you're going to find is when you use the camera, what's in focus for you underneath the eyepiece is not actually going to be in focus on the camera. So you're going to have to mess around with it a little bit. But oh, lo and behold, we have the undersurface of a leaf. And I'm going to get really close so you can see this, how cool this looks. Isn't that cool? The big circular things are the stomata. Okay, so we have this at 40x. You can see that right on the 4x. So what we're going to do now, what you're going to need to do, is to not have the camera fall over, is you're going to need to bring it into focus at 100, ultimately at 100x. So what I'm going to do is I'm going to talk to you, but I'm going to show you what it looks like on the scope itself. So you're actually looking at the computer. You are not looking at me on the scope. And did you see the stomata? They came into focus. This is actually stomata of mother-in-law's tongue, which is a cam photosynthesizer. Uses cam photosynthesis. So we went from 4x and we're now at 10x, so we're at 100 magnification, and we're actually going to take our data, our measurements, our final picture at 400 magnification. So I've just rotated the objective lens to the 40x, and we're going to let that come into focus. The camera's working. You're going to see it's going to be a little bit blurry. So you're going to have to use your fine-tuned focus. Oh, and we don't even have one here. So I'm going to have to move my stage arm to bring one into central view, because the camera's much more limited in its view. Oh, isn't that just beautiful? Oh, nope, I just lost it. There we go. Okay. So that, my friends, is a stomate, a singular stomate on the surface of a leaf of mother-in-law's tongue. And I'm changing the focus slowly here, and that's why it's blurring in and out. You're going to want to get a nice close-up image of a stomata. This is a singular stomate on the leaf surface of mother-in-law's tongue, and ultimately I'm going to want to measure that. And so I am going to want to take a picture of that. So I'm going to come over here, and I'm going to bring the camera up here like this, and I'm going to come over here to snap. And it's going to snap a picture for me, and then I'm going to make sure that I save that picture somewhere. So I might call this one MIT 400X. I'm going to save it, and I will show you in another video how to make a copy of it. All right, so that's how to use a camera with a microscope. Please remember that when you are done, you're going to disconnect the camera from your computer. You're going to remove it from the ocular stage, and you're going to replace that eyepiece. Remember, these are expensive, so you need to be careful with it. Hey, have fun with your stomates today.
All right, see y'all in lab.